So hello all, welcome to our presentation. I'll be talking first today. I'm here with Nikki, who will be introduced later. I'd like to thank Robert at the Biosample Hub for inviting us to share our Living Biobank activities. So currently, I believe we are the only Living Biobank registered at the Biosample Hub. So I'll have this few introductory slides of what the Living Biobank is all about, and Nikki and I will both go through the standard methodologies in establishing and using the patient-derived models. I'll go through the PDX, and Nikki will go through the PDO platform. Next slide. So before introducing the new version of Biobank, I like to introduce the term biobank. This was commonly used starting in the 1990s, so it's not very long ago. And when most biobanks, this was when most biobanks were established, primarily for the purpose of collecting and preserving flash frozen or formalin fixed paraffin embedding of human biological materials for scientific research. For example, here you can see the different types of biospecimen from the surgical suite. We tend to get specimens that are about 1 centimeter in size. And from the biopsy suite, these noodle-like tissue fragments are about 1 to 2 millimeter in width here, and the largest tend to be about 1 centimeter in length. And then we get our fluids anywhere between 10 mil to a liter of fluids. So in terms of our new version, the Living Biobank, we established patient-derived resources, including the patient-derived organoid platform and also the patient-derived xenograft models. And we provide these resources and expertise to the research community, whether it be the academia or the industry partners. Researchers at our institute have been independently establishing patient-derived models for over a decade, as you can see here on the common tumors. And since 2016, our core was formed and we collaborated with PM researchers to consolidate our resources and support investigator-driven science. Models across cancer types have been consolidated for effective maintenance and distribution of the resources. As you can see here, the living aspect of it is to bank the viable tissues, and the inventory itself, we rely on our standard Princess Margaret Cancer Biobank to do the inventory banking for us and the accessioning of our specimens. The living part also, beyond capturing the minimal essential clinical data you can see here, is capturing all the proteogenomics profiling data and also the experimental data, because this helps users to progress in the research and make use of the science that's there on the models. So the Living Biobank itself, between Nikki and myself, we also initiate study to optimize methodologies for integrative use of the PDX and PDO programs. So these methodologies are highly specialized, resource intensive, and time consuming to generate renewable in vitro and in vivo models, which we hope would maintain the high fidelity to their parent tumors. And so far, accumulating evidence shows potentially high clinical utility. So just to step back a bit, this slide gives us a quick review on the different experimental systems for human cancers. And we have the long established cell lines in monolayer cultures on plastic dishes. And some of the advantage of these are that they have fast doubling time, easy to manipulate, and overall, the global community have collected a lot of data and experience. Although they do have limited characteristics that would reflect the original patient tumors. Compared to the 3D organoids, which will be discussed later on in the gel matrix, and also the xenograft platform, in particular, the patient-derived tumor xenografts. These are early passages of tumors in the immunodeficient mouse hosts, and some advantages include that they perhaps mimic better the parent tumor heterogeneity, the tumor microenvironment, and they have a higher success rate of establishment than the in vitro models. Now, do these models mimic patient characteristics, and why do we need so many individual patient-derived models? So in this slide, I like to point out that at the patient level, for example, here at one of the largest consortium, the Cancer Genome Atlas data portal, this website showcases tumor heterogeneity and diversity as observed in the comprehensive multiomics profiling of 11,000 human tumors. So the question here would be, what functions do these genes have in cancer cells, and how do they affect patient management and treatment outcomes? And can we use patient-derived models to study these functions? So here is an example of PDX models being able to mimic patient tumor characteristics. This is a study from our lung program team. In this paper published, here's the PMID, to comparing the PDX genomic profiles to their corresponding patient tumors. I decided to show here the figure of PDX phosphoprotein profiles derived from liquid chromatography and mass spectrometry analysis. And in this panel at the left here, it's a comparison of phosphorylation profile from independent studies of the publicly available data cell lines shown with these pink dots, and patient tumors in the blue, and the normal tissue. And in this left panel, our PDX specimen shown in the orange dots. And you can see here that the orange dots overlap with more similarity to patient tumors than to cell line features shown in the pink cluster here. In the right dendrogram panel, this displays the phosphoprotein for a list of the receptor tyrosine kinases. With the green label PDX lines, you can see marked, embedded with the patient tumors in the blue versus the cell line in the pink. This also shows that PDX have more similarity to the patient tumors rather than to the cell line. So knowing this, do these characteristics translate well to predicting clinical tumor behavior such as drug response? So in this next slide, we show that, when we compare PDX drug responses to the patient tumor, what is happening? In this example, biopsies have been collected along the patient treatment timeline at the hospital. We can see here the two biopsies. The first one is after the ALK inhibitor crizotinib, and the later biopsy after the patient had progressed on these two ALK inhibitors. So this lung tumor was initially diagnosed with the ALK positive test, and the patient was administered to the multiple ALK kinase target therapies all along here. As resistance occurred from the first to higher generation of ALK inhibitors. This resistance was mirrored in the PDX model at these two time points. In the left panel here, you can see here from the first model, PDX model 2A, show resistance to crizotinib compared to the higher generations of drugs where response was detected. So here we can see that initially there was some response to this first ALK inhibitor, and then resistance occurs just as the patient is progressing on, and they had to be switched to the second inhibitor. And here we can see that with the second and a later version of ALK inhibitor, this tumor is still quite responsive. But in the second model, model 2B, mimicking its clinical on-drug tumor progression state, it showed resistance to all three ALK inhibitors, as you can see by these color lines. It's growing. Even though there's some delayed in the response, there's the tumors are definitely not flatline as in the model 2A. So by whole exome sequencing analysis and validation, were we able to find any alterations that would result in this resistance? So the model 2B was characterized by a BRAF mutation absent from this first model at the first timeline collection. Here the team showed that treatment inhibiting the bypass of the BRAF-MEK pathway alone was insufficient to inhibit tumor growth. As you can see here, this reddish-brown line is growing similarly to the vehicle, and that it required a triple drug combination of ALK, BRAF, and MEK inhibitors to overcome the resistance. And here we see a great tumor response in the green line. So this is a highly effective inhibition of tumor growth. So given the highlighted examples, to proceed forward, could we study molecular mechanisms of cell behavior using these more complex patient-derived models compared to conventional cell lines? We know that standard cancer cell lines in preclinical drug testing fail to predict clinical outcomes over 80% of the time. So evidence is accumulating for the utility of these new patient-derived models, but more study is needed on larger numbers of models with consensus on standard operating protocols. Next, I would like to share with you some of our PDX methods for study design. The PDX method itself is quite resource intensive with prolonged time to establish a stable model. The methodology for PDX establishment requires a highly skilled technician for surgical methods and to monitor and administer test agents. And most of them, if it's targeted therapy, would be oral gavage. So the growth cycle demonstrated here from patient to the first implant tend to take anywhere between 2 months to 6 months. And then when it's implanted, the maximum size at which it is harvested is at 1 centimeter in largest diameter, in which case the tumor would be divided and fresh tumor would be, a fragment would be re-implanted into the next generation, which we call passage 1, 2, 3, and so forth. So histologies review per mouse replicate and quality control tests, including identifying failed tumor growth due to lymphoma outgrowth, as you can see here with the tumor islands, or the bacteria contaminated tumors, as you can see in this panel, the bacteria plague. So models are tested regularly for mirroring pathogens and DNA fingerprinting. So this slide shows a typical PDX drug study efficacy drug study design. We tend to use cryo vials, and we implant them into mice in which we call the donor phase. And we tend to use the expansion from fresh tissue into the experiment phase. And you can see here, for example, of a 3-group experiment, 3 different conditions, the vehicle, a standard of care drug, for example, and a novel drug to be tested. Different replicates can be used, and anywhere between 3 to 12 are the mice replicates. And here is an example at closer look of the tumor volume measurement at the subcutaneous flank. Right here, you can see here with our digital caliper. So twice a week, this is being measured, and each of the dots here are the measurements along the days. And we can see implant, it requires around 2 to 3 weeks before we start drug treatment at day 0, in which we get a positive growth slope, and we randomize the mice into their study groups, and dosing starts. And here we can see the treatment group with docetaxel, a standard of care anti-cancer agent, and we can see tumor inhibition and actually decreasing cell death happening into an almost non-palpable form. Oh, there's a question. My question, can you read it out, Nikki? Oh, yeah. Oh, can you please ask your question? We can't hear you.
Hi, Nuh-An.
Yep.
Can you explain, when you say there's a question, do you mean you can see a question on the bottom of the screen, or?
Someone raised their hand.
Oh, someone, no, that's from the very beginning. Please ignore that. That's okay.
Oh, okay. Okay. So as I was just describing here, the growth curves and how we get our tumor measurements from the caliper measurement here. And we have a system set up where it's all digitalized. The weight of the mice measurements are also being captured, and we use the study log mouse database. It's a licensed database set up on our laptops. So we've been able to accumulate data from many studies to optimize for drug screens studies, as reported in this manuscript by Jessica. And the study made use of data collected over 10 years, totaling 44 studies in the lung program, to evaluate the variable parameters of PDX models and addressing how we design our studies. For example, how do we quantify response? As you can see here, these are just predicted growth curves here. I've redrawn them by hand here, and you can see, sometimes there may be a delay in the growth, but it's still growing. And then we have what we call this minimal residual disease phase. If you stop treatment, the tumor, how we know it's still viable cells remaining is that the tumor will start to grow again. And then we have other conditions where you do have total inhibition of tumor growth and cell death. So this paper discusses various parameters and modeling tools we can use to categorize our response. This is important when we do correlations with our genotypes to see what genotype is predicting for what kind of response. And should we increase our mouse replicates? Should we increase the number of measurements per week we do? And should we increase the duration of drug administration? So this is a paper here I've put if you're interested to delve deeper into how do we best design a study. So beyond our lung tumor program, here I show a summary list of the inventory of the common tumors we've banked in collaboration with PIs, initiatives, and their research programs listed here at our institute, the Princess Margaret Cancer Center. And we can see a list of 8 other tumor types. We perform pilot studies to evaluate engraftment feasibility in new cancer types and methodologies with these researchers. Together with this tissue inventory, I also like to highlight the data inventory aspect, which is blooming for both Nikki and myself, and we don't have enough support in this, so we're still learning. Which includes the minimal essential clinical information and also the molecular profiles. So like the tumor atlas presented in the earlier slide for patient tumors, here at our institute, we support a local version of the CBio portal. In this case, it's .ca. We have our own portal for the patient level, as well as the match patient-derived models. You can see here the PDX models. And here we have the latest addition of the organoids being profiled. So this oncoprint here is an example of the visualization that is supported on this portal. So you can select for your models and the gene panels of interest, and it will showcase it as an oncoprint and show the heterogeneity, for example, here across 4 genes of our lung models per column here, right? And this allows researchers to select for specific genotype for their combinations to evaluate the targeted therapies or interventions. And for example, here, one of the graduate students' project was being done on the KRAS mutant models, which I'll show on the next slide. So this slide shows 10 KRAS models that were selected and tested with 2 targeted therapies to different proteins, which may impact on the downstream KRAS mutant signaling. So in this first graph here to the left, shows mouse replicates, these gray lines, across all the treatment groups and the vehicle. And these lines are the pooled result per group, the vehicle compared to selinexor and trametinib. It uses log linear mix effect modeling. And in the right panel, we report this as the weekly changes in growth rates of the treatment compared to vehicle control group. And if you're interested, I would direct you to this paper that's been published on this study. And, you know, one can ask, are there differences in response depending on the subtypes of the KRAS mutations, as you can see here, the G12D, G12C, and so on, and with other tumor burden genotypes, such as being P53 wild type or mutated. So the experience gained from these type of drug screen is that we would much prefer to start with a more effective screening strategy, definitely shorter than 1 year duration, as it required to do this drug screen across 10 models and 2 drugs, if completed by 1 staff. So my question would be, if we could establish organoids, would the xenograft-derived organoids be a faster platform and more cost-effective than the PDX models, which then would be used to verify the in vitro results rather than doing the screen in the in vivo first? So could we evaluate efficacy of more drugs and their combinations? So I would like to stop here for my PDX session, and I would like to use this point to transit from PDX to PDO methods and introduce Nikki. But before we continue, before Robert continues with his introduction of Nikki, are there any questions for the PDX, or it could be left till the end of the whole presentation?
Nuh-An, I'm sure there will be questions, and we can just wait for the questions to come in. We've got 26 participants. Can I just ask one question, really based on a discussion we had before?
Yes.
And that is, I think you'd quite like to see more collaboration. You'd quite like to encourage collaboration regarding PDX models. Could you say something about that?
Yes, that would be based on this slide here. Why would I encourage more living biobanks to form is because, as you can see, across our more than 100, it's not shown here, but we have more than 100 lung tumors. We can see that when we go down to genotyping, we only have several models for specific genotypes of interest. Here, for example, the EGFR mutants, the KRAS, each of our mutants are in the green. They're the center dots here. Sorry, the red bars are amplified genes. So the mutants would be here. So we go down to numbers of, 10 or less per genotype of interest. And then you would look into the burden of the other co-mutants. So depending on your experiment, you can have just one model from like an inventory like ours, which is quite large. So in that case, I would encourage collaborations and sharing of models, specifically when they've been profiled, so we can do more higher numbers of drug screening for specific characteristics.
That's great. Thank you very much. Okay, we have one question. Where are we?
I'll just read it out.
Okay.
How we manage to make a compromise between the size of the tumor, the expected result, and the management of pain in the animal. Yes, so that comes down to, I believe, if you're talking about here. So the size of the tumor, tumor burden is an issue, and the health is being monitored weekly as we're measuring the tumor. So anytime we have criteria in terms of health parameters that we rely on, and we work with the vet to come up with these parameters. For example, their weight, their body weight, it cannot be more than 20% weight loss compared to day 0 here. So we measure twice weekly, and we give supplements, or we really have to stop treatment if there's any signs of deteriorating health for the animals. So the maximum size is about 1.5 centimeter in largest diameter. And if you tend to grow these any larger, you'll have a huge necrotic center core. So that wouldn't be healthy either. It's just part of the limitations of these models that, you know, within, I would say at 1500 millimeter cubic is a reasonable size to look at healthy tissue. So we always take endpoint and do histology review on these and see what is left behind. I hope that answers the question. If not, we can discuss later.
Okay. Well, it looks like there are no other questions. So, oh, there is one other question. Here we are. Okay.
What is the recommended maximum co-ischemia time if we intend to work with viable tissue of good quality? How do you handle the freshly excised tissue? So this is based on practice and how far we can push the system. And it really depends on a few things. Nikki and I, you know, we try to push what we can, what the clinic can extract and then have it delivered by, you know, the coordinators to our lab. Is the quickest is from biopsies, and we get it between 2 hours. But, you know, usually I would say between 2 and 4 hours is a typical time that we get fresh in the lab, and we try to use it on the same day. And for most of the tumors, it's worked well. For the more aggressive tumors like pancreas, I think Nikki's been able to get away with longer, like overnight in the fridge kind of sample handling, right, Nikki? So it depends on the biology of the tumor. But from experience, we know that using the tissue, the fresh tissue from the clinic within the same day is the best. And the sooner, the better.